let's close with BCMA as a target, which I think's got everybody excited. And let's talk a little bit first about antibody-drug conjugates. Faith, have you had any experience with the BCMA antibody-drug conjugate?
So no,
yet?
I haven't. I've been looking at the kind of literature. It looks really very impressive. I mean, when
Tell the audience a bit what's got you excited there.
So in the when Suzanne presented the initial data, 60% response rates with the antibody-drug conjugate, and very good tolerability. I think there's a few unusual side effects attached to it, which, just as with all the other drugs, we're going to need to learn how to manage and moving forward and so on. But it seems to be that it works very well, and this whole concept of having your kind of payload on your antibodies seems to be opening up.
It seems to validate BCMA as a target.
Definitely, definitely.
We are just opening the clinical trials now, but have anybody had actually an opportunity to use the drug?
We're opening it as well.
Same thing.
Just looks very exciting. 60% response rate in fairly heavily pre-treated patients, and some corneal toxicity, but otherwise people are getting through it. It leads us into, so BCMA is now validated as a target thanks to that drug, and there's some very exciting data being presented at this meeting about a BiTE, a T-cell engager molecule from Amgen actually, but there are others out there. There's probably a dozen of these drugs now in clinical trials. We haven't seen the updated data, but in the abstract it looks like there's some very deep responses with an anti-BCMA BiTE, which of course I think in that study was a continuous infusion, but they're now being studied as a weekly dosing schedule. So anybody want to take a crack at their enthusiasm for that?
Sure. So BCMA, 420 is the study we're talking about, short-lived. As just escalation, as they get into the higher doses, short-lived half-life for the antibody. But as they get into high doses, they're seeing CRs and MRD negative status. So I think the proof of principle is there, and that's why there's so much excitement, even with a phase 1 trial. BiTEs, potentially could be a major competitor to CAR T cells because, like we engineer these T cells to recognize the cells, and I describe the BiTEs. There's like just double-sided Velcro straps. On the one side you have the myelomas, on the other side you have the T cell, but they're off the shelf. You can start treating a person, the day you decide to start treating.
Faith, you excited about the BiTEs?
Yeah, so we're involved in the once-weekly study, which hasn't reported its data yet. But I think that continuous schedule is great. There were, as we see with the CAR T cells, again, unusual set of side effects, so we have to be careful with the CRS and so on.
I haven't read the abstract carefully enough. What does it say in there about cytokine release syndrome?
So it does occur. So, I mean, it's manageable, okay, but it definitely occurs, and I guess that's that
part
whole
of the
question.
Magic of these therapies.
I was going to say,
manageable.
I'm not sure you get a response without, there's always some form of toxicity, unfortunately.
So pretty early days, but it's like this sort of the equivalent of blinatumomab for acute leukemia. It looks like we've got some of those drugs coming.